Hello students, in this lecture we shall discuss the topic the structure-function relation in proteins. By now you are well aware that the structure of a protein determines its function. We shall understand this concept by taking the example of a proteolytic enzyme chymotrypsin, which is secreted into the duodenum of the small intestine for digestion of proteins in the food we have ingested. Chymotrypsin is a globular protein and its 3D structure shows that it is made up of three different chains. This chymotrypsin is a very strong peptidase. It is synthesized by the pancreas and is sent into the duodenum through the pancreatic duct. However, it does not harm the cellular proteins of the body itself. How? Nature has ensured that chymotrypsin and the other proteolytic enzymes are synthesized as inactive harmless precursors known as zymogens, which are activated later on when required, that is, only in the duodenum. These zymogens are inactive because their substrate binding sites are blocked. When these reach the duodenum region, first the trypsinogen is converted to trypsin. This active enzyme then brings about a proteolytic cut in the trypsinogen, which results in a conformational change, exposing the substrate binding pocket and converting it into an active chymotrypsin. This phenomena is known as in situ activation. Let us now understand the mechanism of proteolytic action of chymotrypsin. But before that, we need to understand the structure of this enzyme in depth. A full-featured 3D structure viewer is available on the site of NCBI. This beautiful structure clearly shows the alpha helices, beta sheets, and the unfolded regions of the polypeptide. At the center, you can clearly see a small pocket. This pocket is known as the hydrophobic pocket and serves as a substrate binding site. A close-up of its structure shows that the substrate binding site contains a catalytic triad, that is, a group of three special amino acids arranged in a particular specific conformation. This triad consists of aspartate, histidine, and serine, which together break the peptide bonds of the protein substrates coming into this hydrophobic pocket. Chymotrypsin is a serine protease, as the main role in the catalytic mechanism is played by the serine residue. The most important point to be noted over here is that the triad is made up of aspartate 102, histidine 57, and serine 195. The numbers indicate their positions within the primary structure. As you can see, they are widely separated from one another in the primary chain structure, and yet when the enzyme adapts its final tertiary fold, these amino acid residues are perfectly positioned within the active site and should be able to bring about the desired catalytic reaction. Thus, this example is the perfect demonstration of how protein structure is so vital for its function. In this triad, the serine provides a nucleophile as there is an oxygen atom. Histidine acts as a base catalyst to activate serine. Aspartate plays a supportive role and stabilizes the protonated histidine. Overall, the reaction mechanism is one of the covalent catalysis, which involves a two-step reaction where the enzyme actually forms a covalent bond with its substrate. Let us now understand this triad in detail. So we saw that in the 3D structure of the enzyme, these three important amino acid residues come close together in space, which allows a charge relay system to operate. That is, the negatively charged aspartate 102 is able to hydrogen bond with the adjacent histidine 57, partially borrowing the hydrogen ion from the latter. The histidine 57 compensates its hydrogen loss to aspartate by attracting hydrogen ion from the adjacent serine 195. So this looks like a relay race where the charge is being passed on from one member to the other. A very important point to be noted over here is that not all serine residues of the chymotrypsin protein are acidic in nature. It is only the 195th serine which is acidic due to this special conformation. the first thing that will happen is that the histidine will act as the base catalyst and extract a proton from the serine. So here's our electron-rich nitrogen atom, which will attract a proton and form a new bond with hydrogen. As it does so, the hydrogen will break its bond with the oxygen, and that allows the oxygen atom to form a new bond with the carbonyl carbon. So here we see that nitrogen has formed a new bond with hydrogen, and the bond between oxygen and hydrogen has broken. But it does form a hydrogen bond, as shown by this dashed line. The oxygen in the serine has formed a new covalent bond with the carbonyl carbon of the substrate, and this is basically an unstable tetrahedral intermediate carbanion. In the next step of the reaction, the histidine is now going to act as an acid catalyst. Remember, it has an extra proton it can donate. Only instead of donating it to serine, it is now going to donate it to the nitrogen of the sessile bond. Sessile bond means the bond that is going to break, this one. In doing so, the nitrogen-hydrogen bond will break. Nitrogen will receive its electrons back and will become neutral again. The free hydrogen will form a covalent bond with the nitrogen of the peptide, and this would result in the breakdown of the carbon-nitrogen bond of the peptide. Hence, the substrate that has entered the hydrophobic pocket will leave in two fragments. The C-terminal fragment, this one, and the N-terminal fragment, this one, will separate out. The C-terminal will be released free, but the N-terminal will be held to the serine residue for some time. This histidine is back to its original form, but we have a covalent intermediate which has to be separated. Now, in the next phase of the mechanism, the previous series of steps will repeat themselves in the same fashion, but the nucleophile here will be different. So again, we have histidine acting as a base catalyst, but now our nucleophile is a water molecule. And so histidine is going to extract a proton from the water molecule, forming a new nitrogen-hydrogen bond here, and this will allow the oxygen in the water to act as a nucleophile and to attack our carbonyl carbon here. So this step is the exact repeat of the earlier step 1. Now that we have a new nitrogen-hydrogen bond here, histidine will carry that positive charge, which is stabilized by the adjacent aspartate residue through this hydrogen bond here. Now we have a carbon-oxygen bond which forms a new tetrahedral intermediate unstable carbanion. Finally, histidine donates the proton back to serine, and so serine releases the carbon of the intermediate, which now carries the OH group of the water molecule. This renders the active site just as it was before the whole reaction began. So the enzyme has been regenerated, and the N-terminal portion of our substrate peptide is released after the C-terminal portion. This is how a chymotrypsin enzyme breaks the peptide bonds one by one of the incoming proteins. This whole mechanism demonstrates that how the correct folding of the protein will result in generation of the correct domains or the substrate binding sites and will be responsible for the catalytic reactions. That is, the structure of the protein is totally responsible for its function. Thank you.